Synaptic transmission is the process of transmitting information from one cell to another across a synapse. There are two broad categories of synaptic transmission: chemical and electrical. During chemical synaptic transmission, the presynaptic neuron releases neurotransmitter that crosses a synaptic cleft and binds receptors on the postsynaptic target. This is a unidirectional process. During electrical synaptic transmission, an electrical signal passes directly from one cell to another through a gap junction. This is a bidirectional process.
Spiking activity is shared by neurons through two major modes of synaptic transmission: chemical and electrical. In both cases, there is a presynaptic side formed by the axon terminal of one neuron and a postsynaptic side formed by the dendrite of another neuron. However, there are key differences in how each mode operates. Chemical neurotransmission relies on the snare complex, which helps dock readily releasable pools of synaptic vesicles in the active zone and initiates vesicle exocytosis, releasing neurotransmitters into the synaptic cleft. Voltage-gated calcium channels are located in the active zone and provide the source of calcium necessary to trigger this process. Vesicles are then recycled and refilled after use. The coupling of voltage-gated calcium channels to the snare complex elegantly links activity levels to neurotransmitter release. As presynaptic activity levels increase, more spikes invade the axon terminal, activating more voltage-gated calcium channels. This allows more calcium to flow in, which more effectively activates the snare complex. This in turn leads to more vesicles fusing and more neurotransmitter release, which translates into stronger activation of postsynaptic receptors and thus more effective communication. Neurons can either excite one another using acetylcholine or glutamate and increase spiking activity, or inhibit one another using glycine or GABA to decrease spiking activity. There are two major types of receptors that so-called excitatory and inhibitory neurotransmitters activate. Ionotropic receptors contain ion channels that allow direct current flow and so provide a fast means to alter the postsynaptic membrane potential in response to neurotransmitter release. Metabotropic receptors are coupled to G proteins that lead to secondary effects on neuronal excitability and thus are slower in time course. Electrical neurotransmission relies on electrical synapses formed by gap junctions that directly connect the pre- and postsynaptic neuron. Thus, spikes arriving in the axon terminal can move directly to the dendrite, with the amount of current flow limited by the number of gap junctions. In contrast to chemical neurotransmission, both subthreshold and suprathreshold signals can pass through gap junctions. Thus, electrical synapses are faster, more reliable, and bidirectional forms of communication between neurons.
In introductory classes, you learned about synaptic transmission, but neuroscience is an interdisciplinary field, so there are many words for this term. For example, it might also be called synaptic communication. Ultimately, the effect of synaptic transmission depends on how the postsynaptic target is affected. Synaptic transmission is excitatory if the postsynaptic cell becomes more likely to spike, and it is inhibitory if the opposite occurs. So synaptic transmission is an umbrella term that includes both excitation and inhibition.
Synaptic transmission is important in almost every aspect of neural communication, including sensation, motor control, and cognition. For example, did you know that many poisonous substances produce toxic effects in the body because they disrupt the process of synaptic transmission? A tetanus infection is caused when a puncture wound becomes infected by Clostridium bacteria that are commonly found in dirt and rust. These bacteria produce toxins that target inhibitory spinal cord neurons and destroy the synaptobrevin protein, which is part of the snare complex. Now recall that the snare proteins are required for docking and exocytosis of synaptic vesicles. When the snare proteins are selectively destroyed in inhibitory neurons, this leads to an overall increase in excitation that produces extreme muscle contractions called tetanus. In extreme cases, a tetanus infection produces muscle spasms that impair breathing and can even break bones. Treatments include mechanical ventilation to assist breathing and muscle relaxants to treat the muscle contractions.
Now that you've had a bit of review on synaptic transmission, let's see if you can follow along with this example. Above the line on the top of this diagram, I'm illustrating a chemical synapse. We have the presynaptic neuron here, the postsynaptic neuron here, and we have recording and stimulating electrodes in each one of those cells. So here, what I'll be illustrating with plot 1 is the voltage that we record from the presynaptic neuron. And here what I'll be recording on the second plot is the voltage recorded in this postsynaptic neuron. Similarly, on the bottom of this diagram, there is an electrical synapse. And there is a stimulating and recording electrode in each one of the two cells. So again, here I'll use the upper plot to show the voltage recorded by electrode 1, which is in the cell on the left. And I'll use the bottom plot to illustrate the voltage recorded by electrode 2 in the cell on the right. So the question is, given this setup, if we had identified a synapse, but we weren't sure if it was a chemical synapse or an electrical synapse, what sort of data might we collect to be able to figure this out? Well, one of the things that you could use is a combination of subthreshold and suprathreshold depolarizations. If we investigated neuron 1 and we had a chemical synapse, what we would find is that when there was a subthreshold depolarization, we would get an increase in voltage, but no action potential. And when there was a suprathreshold depolarization, we would get an action potential. If recording from cell number 2 under those conditions, when cell 1 did not fire an action potential, it did not release any transmitter, and therefore we would not see any voltage change in cell number 2. Assuming that this is an excitatory synapse, when cell number 1 has an action potential, we would see an EPSP, an excitatory postsynaptic potential, in cell number 2. Had we used an identical stimulation protocol, but had been investigating an electrical synapse, we would find the following. In cell 1, we first start with a subthreshold depolarization, and then also a suprathreshold depolarization. And if we were recording from cell 2, what we would find is that the voltage that we see in cell 1 can spread directly to cell 2, even if it is subthreshold. So we might see a very minor increase in voltage in cell 2. And also, when cell 1 is spiking, that voltage might trigger, similarly, an action potential in cell number 2. So the key difference here between these two conditions is that in an electrical synapse, subthreshold depolarizations will be communicated from one cell to another, but in a chemical synapse, they would not.